Okay, so we're talking about communicable diseases first. So this word, communicable, remember that you can catch these diseases. They can be passed on from person to person. Remember that communicable diseases are caused by organisms that we refer to as pathogens. So things that cause disease are called pathogens. We can split them up into different types. So the main two things that make us ill, we need to know about bacteria, we need to know about viruses, and then we're going to add to our list diseases caused by funguses and diseases caused by these organisms called protists. And we will have some examples of those diseases. Okay, so you must know that bacteria, when they get into your body, make you ill. All of these pathogens will reproduce inside your body. Bacteria get into your bloodstream and they make you feel ill because they produce toxic chemicals that we're going to call toxins. And the two diseases which you need to know about that are caused by bacteria are salmonella and gonorrhea. Oh, tricky to spell. Let's get it right. One O, two R's here. Okay. Viral diseases, we should know about HIV infection. We must know about measles, and we need to know that there are plant viruses, things like tobacco mosaic virus. Okay. Fungal diseases, in plants, you've got that disease called rose black spot. Humans can get fungal diseases as well, things like athlete's foot, which is very communicable. It's easy to catch. If you're walking around in bare feet in the gym changing rooms or in the swimming pool changing rooms, that sort of thing, you can pick up the fungus that causes athlete's foot. And protist, the example of a disease caused by a protist is malaria. Remember, malaria is caused by the malarial protist, but is carried by mosquitoes, and we talk about mosquitoes as a vector, the thing that passes the disease onto you. Okay, and you should be able to talk about the symptoms of these diseases. So learn the basic facts about the diseases. For example, measles gives you a fever and a rash. Okay. We should be happy to talk about how we can reduce the spread of these diseases, which is very in the news at the moment. Okay, reducing the spread of diseases that might be spread by people coughing and sneezing and touching things. Okay, so we can talk about things like droplet infection. Okay, when somebody sneezes and you breathe in the air, okay, then that's how coronavirus is spreading around, and we can avoid that by wearing a mask, for example. Okay, we might destroy the vectors, we might kill the mosquitoes. Okay, we might get rid of the rats which have the fleas which carry plague, for example. Right, we might talk about isolation in the news, quarantine. Yeah, keep infected people out of the way so they can't spread the communicable disease. And finally, what everyone hopes for, a successful vaccination program. okay, that we're going to do more about in a minute. Okay, the whole point about vaccination, there's two things about vaccination. One, that it stops you from getting the actual disease, and two, that it prevents the spread. So in the news you might have heard about herd immunity. We need to get a certain proportion of the population vaccinated to give us herd immunity against the coronavirus. Okay, and if we get diseases inside us, then we need to defend ourselves against them. Okay, so I'm just going to get rid of that symptoms word down there. Give myself a bit of space. Whoops. So let's talk about defenses, body's defenses down here. We've got two lines of defense for our communicable diseases. We need to talk about prevention. How do we stop these pathogens getting in? And we need to talk about cure. How do we get ourselves better if they do get in? Okay, so to prevent, remember what we said, we need barriers. We need ways of preventing the entry of pathogens. So our biggest prevention is our skin. We've got a layer of it over nearly our whole body. Okay, where there's gaps in your skin, so obviously you've got your nose, your mouth, if you cut yourself, okay, anywhere where there's an entry into your body, right, you need some extra protection to stop bacteria getting in there. So we've got things like mucus providing cells that's sticky and traps the pathogens. Your stomach makes acid to kill bacteria that you swallow with your food. That would be a line of defense. You've got hairs up your nose to try and prevent pathogens from getting in. Okay, and if you cut yourself, your blood clots to seal up the hole and to try and stop lots and lots of pathogens from getting inside you into your bloodstream. Okay, your skin is actually slightly antimicrobial, as are your tears, which protect your eyes. Okay, so a few ways that we defend ourselves. If stuff gets inside, we need to be talking about white blood cells. And three key words to go with that. White blood cells protect us by engulfing pathogens, okay, spreading around them, surrounding the pathogen and dissolving them. Yeah, the posh word for that is phagocytosis. They make two antitoxins to get rid of those poisonous chemicals that make us feel ill, and antibodies that are the chemicals that give us immunity once we've made them. Okay, and the antibodies links to how we explain how vaccines work. Vaccines work by triggering the production of antibodies. Okay, and remember that that production of antibodies is triggered by the presence of these specific proteins on the pathogens, which are called antigens. So there's lots of words that are similar, don't get confused. Antigens are like a little name badge for the pathogen. When they get inside your body, or if you inject the dead or weak form of the pathogen, the antigens stimulate your white blood cells to make the correct antibodies that fit on them. Okay, so a specifically shaped protein molecule. Okay, so we're looking in our chapter B6 in our textbook. Okay, big explanation. Make sure you're really confident about this. how do we explain the process of vaccination and how it works. Okay, let's split it up into bullet points. Okay, what's in the vaccine? A dead or a weak or an inactive pathogen is injected into your body. It stimulates your white blood cells. Once the white blood cells are stimulated, they're stimulated to produce antibodies. So it's like giving a set of instructions to your white blood cells, fooling them into thinking that you've been infected with a pathogen and making them produce the correct antibodies. Right? Now this is the key thing. Because you've done that, okay, if the same pathogen that's live, so if you get the disease for real, enters your body, the white blood cells can make those antibodies really quickly. Okay, so it doesn't stop you from the pathogen entering your body. It means that you can respond really quickly and therefore preventing you from getting ill. So if you've been vaccinated against measles, you will have had the dead measles virus injected into your blood. Your white blood cells have recognized it as a pathogen, recognized that they can make antibodies that fit the antigens on the measles virus. And then if you do get infected with the measles virus, the white blood cells make the correct antibodies for measles fast enough so that you don't get ill. Okay? If enough people are vaccinated, then we can acquire herd immunity, because the spread of the disease in the population will be low enough to stop it being a problem. And that's what we're hoping is going to happen with the with COVID-19 virus vaccination. Okay, right, let's talk about different treatments, and we're going to split drugs that we take to make us better into things that relieve symptoms, okay, and things that are a cure, things that kill pathogens. And our example of painkillers are things like aspirin, paracetamol, Nurofen, morphine if you want a really strong painkiller. And these drugs aren't making you better, they're just reducing your pain. Okay? They treat the symptoms, they do not make you better. Okay? Whereas curative drugs treat the source of the disease, and our example of curative drugs are antibiotics. Antibiotics make you better because they kill bacteria, they kill pathogens. Okay? And that's a way that you actually... Okay, now, these antibiotics, remember, will only kill bacteria. So it's important to realize that they don't work on viruses. You can't treat a viral disease with antibiotics because they only kill bacteria. And it's very, very difficult to make antiviral drugs because of how viruses reproduce. So antiviral drugs are a challenge, and the reason they're a challenge is because the viruses are inside cells. They're hiding. They reproduce inside your healthy cells until there's lots of copies of the virus and then the cells burst open. Okay? So making antiviral drugs is difficult because viruses are inside cells, and so if you have antiviral treatments, they tend to damage your cells as well. It makes it very hard. And when we're talking about making drugs, yeah, it's very important for the upcoming test that you are going to be able to talk about how we can make new drugs, develop new drugs, and be confident that they're safe. Okay, so I want everybody, please, to make sure that you look carefully at sections in your textbooks B6.3 and B6.4. Okay, and we're talking about how do we go from an idea of a treatment to a drug that's available for your doctor to prescribe. So lots of these chemicals that drugs originally came from plants. So a good example of that is foxgloves, yeah, which their Latin name is digitalis. Foxgloves gave us heart treatments. And another example would be salicylic acid, which comes from willow bark, which led to the development of aspirin. Okay, so there's lots of chemicals out there that might be suitable for making new and better treatments. And when you're talking about testing drugs, okay, we need to follow a certain time frame, and you've got 3 key words. When we're testing drugs, we want to know, are they toxic? Okay? Do they work? And how much do I need? Okay, so these 3 things are what we want to end up knowing about by doing tests on these chemicals that might have come from plants, they might have been made in a lab, and we're trying them out and trying to make new and better medicines for people. Okay, so once you discover a chemical that might be a possible drug, okay, the time sequence starts off with taking those chemicals into preclinical, before clinical trials. And here we're going to be testing the drug on animals or tissues and testing for toxicity. Is it poisonous? Yeah, we might be getting some ideas about efficacy, does it work, and dosage by looking at animals and tissues as models of humans. Okay, we'd want to be fairly certain that these drugs are quite safe in terms of not being really poisonous. Okay, before we move on to clinical trials using people. Okay, and clinical trials are split up into phases. Okay, so to start with, you'd always start on a very small sample of people. You'd want healthy volunteers without any illnesses, and you might look for side effects. If there's horrible side effects, we might go back to the drawing board and try one of our other chemicals that we think might be a suitable treatment. Okay? As we get more confident, we might start to narrow down the number of things we're testing. We might increase our sample size. We want lots of different groups of people, ages, genders, races, backgrounds, all sorts of that before we go on to clinical trials on ill people. And once we start treating ill people with our new medicine, we're seeing if it works, and we're going to be deciding on the final dose. Okay, so eventually, after quite a long time, so it might be 12, 14 years before this drug goes from an idea of a chemical to a drug that's in the marketplace, in the chemists. Okay, make sure you're happy about what these 3 words mean. Make sure you're happy that first of all drugs are tested on animals or tissues, as models of people before we risk testing them on humans. And we would start our test with a small number of people in case the drug has horrible side effects. and those people would be healthy. We'd test it on larger samples, maybe with a wider variation in age and background for people. And then finally, we'd be having clinical trials on ill people to see if these drugs actually are better than existing treatments. Okay? Once that's happened, the scientists that are doing the research will have to publish their results. Okay? And we would say in the publishing of results that we would be looking for their peers to review their data. That means scientists that are independent of the people publishing the work are going to look at the data, are going to replicate experiments, and also hopefully confirm that the drugs are suitable to go into circulation. All right. In these clinical trials, remember we use the words double-blind to mean a clinical trial where neither the person giving the drug or the person taking the drug knows whether it is the actual drug or a fake version, a placebo. So clinical trials would usually involve a double-blind placebo trial, and that means we've got a group to compare to. So imagine testing a drug, we'd split up into two groups. We'd try and match the people in the group. We'd give one group the placebo, one group the actual drug, so we can compare them. And we'd be careful that the groups were matched for age, gender, and all the rest of it. The bigger the sample, the more reliable our data becomes. Sure, we're happy about what this word, so non-communicable disease, actually is the biggest causes of disease in the country. We're talking about diseases that you cannot catch. They're not infectious. They're not caused by a pathogen. Okay, and the big non-communicable diseases we talk about are things like heart disease, various types of cancer. Okay, and in this topic you are appreciating that there are risk factors in your lifestyle for developing these non-communicable diseases. Now, these are the biggest killers, in our society, and you can reduce your risk by adjusting your lifestyle. Okay, so we're talking about things like your diet, how much you exercise, whether you smoke, whether you drink alcohol. Okay, and all of these factors combine, including some genetic risk, to give people information about these non-communicable diseases. We might add diabetes to here is a big non-communicable disease problem, especially type 2 diabetes, which is on the rise. Okay, so some details here. So things like poor diet, which also leads to being obese or can do. Obesity is a risk factor in all of these non-communicable diseases. Okay, and obesity links to poor diet, poor exercise. People who are less active might be more likely to smoke. Okay, and you've got a bit of detail in this chapter on symptoms of heart disease and treatments that we can give to people. So, for example, we can reduce your blood cholesterol, which is a risk factor for heart disease and stroke. We can treat that with statins. We can prescribe people access to dietitians, gym membership, improve people's lifestyle. Okay? When you are revising the section on cancers, make sure you are aware of the fact that when we talk about cancer and tumours, that tumours can be benign and tumours can be malignant. Make sure you revise the detail about that. Okay, a benign tumour, right? All cancers are where cells are reproducing in an out of control way.